Alright, so in this video we're going to talk about the mechanisms of antibiotics. Now remember, for antibiotics they can be bacteriostatic or they can be bactericidal. This is the proper spelling of it. I spelt it wrong in the PowerPoint earlier. So let's go back and remember a little bit about our bacteria. So keep in mind that they have their cell membrane, alright, really important to keep all the parts of the cell inside. Alright, and then outside that we've got a thicker cell wall. Right, I'm going to try to do this a little bit neater. I've practiced this lots of times, but I'm guaranteeing you guys I'm going to get it wrong or something wrong, or I'll mess up when I'm trying to talk. Okay, so this is our thick cell wall around the outside of the cell membrane. Okay, so, or thicker cell wall around the outside. Okay, and then remember, with some bacteria, so the ones that are gram-negative also have that LPS layer around the outside. Okay, so if they're gram-negative, they're going to have that fatty lipopolysaccharide layer around the outside. All right. Now, the other thing to remember for bacteria is they have things inside of them as well. All right, so I'm going to go through some of the processes that are in here. So we've got bacteria that need to make folic acid. All right, now humans can't make their own folic acid, but bacteria can. They use this substance called PABA, and they do a series of steps to form it into folic acid. Now, that folic acid is really important for them to be able to form their double-stranded DNA. Now, I know the DNA is in a circular form, but I'm just going to put it in straight form now, just so that it's a little bit easier for our diagram. Okay, so here's our double-stranded DNA. Now, if you remember back to biology, your DNA is the set of instructions for everything that happens in this cell. So we don't really like to let our original set of instructions out because something could happen to them. So what we do is we change or transcribe them into single-stranded RNA. Okay, so we've got our single-stranded RNA here. Okay, and then what we do is we're going to translate that single-stranded DNA into our proteins. All right, so we've got proteins here, and the proteins can make up structures for the cell, or they can be enzymes that instruct the cell of what to do. So the proteins are really important here. Okay, now in order for those proteins to form, we need to have these little guys called ribosomes, who are the factories. You got a little small subunit and a big subunit on the ribosomes. So they take the amino acids and they help link them together in order to make the proteins. All right, so that's basically where these are the kind of things that we can affect in the bacteria cell. So let's go through. Now I'm using my notes, so I know I got two notes here, and if you know me, we know we're going to be all over the place here. All right, so let's just start with the first one. So if we have a gram-positive bacteria, so gram-positive bacteria is not going to have this LPS layer around the outside. So if we've got a gram-positive bacteria, what we can do is we can disrupt this cell wall around the outside. Okay, so let's do something to disrupt the cell wall. Okay, so let's disrupt the cell wall. Alrighty, so basically what we're going to do is we're going to prevent the sugars from being able to link with each other. So the sugars will be there, but they won't link to each other. So stuff can leak in and out of the cell pretty easily. The cell will end up with so much pressure on the inside that it basically ends up bursting open. All right, so this is going to be a bactericidal. We're going to wreck this cell wall. Sorry, I got that kind of down there on the cell membrane. We're going to ruin this cell wall. All right, so we're going to destruct the cell wall. So we're going to be able to use this for I'm going to do this another color. We can use this for gram-positive bacteria. Alrighty, so we're going to ruin the cell wall synthesis. Cell wall synthesis. So it can try to put the pieces of sugars together, but it won't be able to. All right, so what ends up happening here is the cell bursts open. All right, so we've basically killed that cell. All right, so this is stuff like your penicillins. So this is what penicillin does. And if you've ever used, I don't know if it's called bacitracin or bacitracin, all right, this other antibiotic. And again, I'm not going to ask you the names of the antibiotics. It's just kind of neat if you get penicillin. You can just say to yourself, oh, that's a gram-positive bacteria that I'm being treated for, and we're busting open the cell walls, or we're preventing the cell walls from being put together properly. Okay, so the other thing we can do here is we can kind of ruin this LPS layer, or we can ruin this cell membrane. Okay, so let's go through there and let's ruin the cell membrane function, okay, of that, or the LPS layer. Okay, so we can use the cell membrane. So we're going to disrupt the structure and function of cell. I'm just going to put MB for membrane, or the LPS layer. All right, and this is going to be important. We're going to use this for our gram-negative bacteria. So we're going to use some of these bacteria, or we're going to use some of these antibiotics for our gram-negative bacteria. So remember, we said before, it's really important to know what type of bacteria you're working with, because you can pick the proper antibiotic to treat it. All right, so if you're infected with a gram-positive bacteria, you want to use a antibiotic that's going to, like your penicillin, that's going to ruin the cell wall synthesis. And if you've got a gram-negative bacteria, you want to use something that's going to disrupt the cell membrane, or that LPS layer. Okay, so what we're going to do here is, what these guys do is these antibiotics are going to bind to the phospholipids. So they bind to the phospholipids, all right, that are in the cell membrane. So the phospholipids. Now, you may say, hey, we've also got phospholipids in our cell membranes, but what they do is they bind to special phospholipids. So there's some phospholipids that are in the bacteria cell membrane that the human cells don't have, so they've made the antibiotics specifically go onto those little special phospholipids. They're called phospholipid 2, or lipid 2. So, but just make sure you know that they bind onto the phospholipids, all right? So, they bind onto those phospholipids in both the cell membrane and to the LPS layer, all right, which again disrupts that cell membrane. So what ends up happening is the cell starts to get really, really leaky. The inside of it starts to leak out, and the cell basically ends up dying. So this is another bactericidal. So these two guys here are both cidal. So this is going to kill, and this one's going to kill outright. Okay, so those two things we're gonna kill our bacteria cells doing either of these things. Now, this would be your polymyxin. So if you ever have a polymyxin, polymyxin, that is your antibiotic that's used to kill off your gram-negative stuff. Now, just while it's in my mind, polymyxin, if you've ever used something like polysporin, all right, which is, you can just buy over the counter, polysporin, and it's the kind of thing you put onto cuts. It has antibiotics in there that will treat both gram-positive and gram-negative bacteria. All right, so the polysporin has a mix of antibiotics. So I'm going to put a mix of antibiotics to treat gram-positive and gram-negative bacteria. Okay, so, and those are usually, you use a polysporin for infections that aren't too serious. All right, so the next thing we can do here is, we just said how important it is for the bacteria to make folic acid, so the folic acid can help form the DNA. Okay, so let's be bad and let's do something. If you've ever had sulfa drugs, all right, the sulfa drugs, those in here ruin this pathway. There's a couple other antibiotics as well, and there's a couple steps in this pathway, but we're basically just going to worry about going from PABA to folic acid. All right, and keep in mind again, humans get folic acid from our food, so we don't have this process in here, so we can take the antibiotics and it's not going to bother us because we don't even have this pathway, so we don't need to worry about it at all. Alrighty, so, well, let's just put a little thing in here. Okay, so folic, we're going to ruin our, what color am I using? Blue. We're going to ruin our folic acid synthesis. Okay, so we're going to ruin that for the bacteria. Sorry, I keep moving this sideways so I can write better. Alrighty, so bacteria, I'm just going to put back for bacteria, can make folic acid on their own. All right, so just so you remember, we have to get it from food, but these guys can just make it on their own. All right, so what happens with your sulfa drugs is we're going to stop the enzymes. Okay, because remember, the enzymes are proteins. So we're going to stop the enzymes in here. We're going to stop this process. We're going to wreck this process there. All the little chemicals that help change that PABA into folic acid, we're going to stop them from working. All right, so we're going to stop the enzymes from converting PABA to folic acid. Okay, now if you have a bladder infection, this is the kind of antibiotic they'll give you for this. So this is your sulfonamides. I spelled it the American way. That could be a PH if you're doing it Canadian or British. All right, so we're going to use our sulfonamides in there. The bad thing about this is a lot of bladder bacteria have become resistant to the sulfonamides. And remember, a lot of times with bladder bacteria, you want to treat this quickly because you don't want to have them have enough time to form a biofilm on your bladder, which makes it really difficult to treat. Okay, so one of the things we can do here too now is, so we've wrecked our folic acid so the bacteria can't make proper DNA. So this bacteria itself can stay alive, but if it wants to reproduce, all right, or it wants to make some more proteins or it wants to grow or whatever, it won't be able to because now it can't make the DNA properly. All right, so it will eventually just die. All right, so the next thing that we can do here is we can destroy our structure and our function, structure or our function of our DNA. Structure or function of our DNA. All right, because we've got this is our original copy of all the information that the cell needs to live. So if we can destroy that somehow or prevent it from being able to replicate, then we'll knock this guy out. So if you've ever been prescribed something like Flagyl or the Macrobid or the Cipro, this is that type of antibiotic. Okay, I'm going to put them in here, but again, you don't need to remember them, but just if you're ever prescribed them, it's kind of cool to sort of know what you're being prescribed. So the Flagyl, the Macrobid, or the Cipro. Okay, that's these type of antibiotics are going to do this job here. So two things can happen here. Either we're going to affect the nucleotides that are in here, so we're going to affect the nucleotides. So if you remember, your DNA is made up of a number of nucleotides that are all linked together. So we're going to affect the nucleotide, or we're going to make the DNA just fall apart because it has to be double-stranded and then it falls apart when it needs to be falling apart to open to for making the DNA or for replication. So what we can do is we can also affect the DNA gyrase, which is important to untwist it. All right, so those are the two ways. So we can either go in there and just kind of like bash up the nucleotides, or what we can do is we can cause it to become unraveled, all right? It has to stay nice and raveled until it's needed to become unraveled. Now, the other thing we can do is we've got an enzyme that's in here that takes the DNA and changes it into RNA, and it's called RNA polymerase. So the other thing we can do in here, okay, we can target this enzyme that's here. This is called RNA polymerase. All right, and you might be able to hear my neighbor chainsawing down his freaking forest. All right, so anyway, we've got this DNA polymerase is a really important enzyme that takes this DNA and changes it into this RNA or transcribes it into the RNA. Okay, so if you've ever been prescribed something like rifampin, all right, that's an antibiotic that's going to make this guy not work. Okay, so we're going to stop or prevent or let's put stop RNA polymerase. Okay, so it binds it up so it can't do this job in between these two things. Okay, so that's rifampin. I might be saying these antibiotic names wrong. I just say them how they're kind of spelled. All right, so we can go through, before we do our last one. So we can ruin the cell wall completely, don't let it get put together, and then the cell ends up with so much pressure on the inside it virtually explodes open, so we've killed it. We can kill our bacteria by ruining the structure and function of the cell membrane or the LPS layer. So we're going to bind to those phospholipids, which makes the cell really leaky, and then it just kind of like leaks all its insides out, and it just ends up dying because it's all leaked out. We can destroy this is a static, okay, because it is going to the bacteria will still be alive, but it won't be able to make the folic acid anymore, and we need the folic acid to make the DNA. All right, or we can ruin the DNA structure. We can either like knock out those nucleotides, or we can cause the DNA to become unraveled so it can't properly be transcribed into RNA. All right, so we can unravel it here. This is also another static, so it's alive. It just can't reproduce properly or make proper proteins. We can also take the RNA polymerase. This is going to be another static thing here. Take the RNA polymerase and block that up so it can't do the job of taking the DNA and then making the blueprint RNA from that. So our last thing here is we can actually go in there and mess up the protein synthesis, okay? Because we need those proteins. Those proteins are going to form the structure of some of the cell itself. It's going to make the enzymes that tell the cell what to do, all right? So we can disrupt, or prevent, I should say. Let's say prevent protein synthesis. Synthesis. Okay, now remember we talked about those ribosomes, and we said the ribosomes in a bacteria or cell are different than a human cell. So this time we've got the 50S ribosome, which is the big one, okay? This is going to be targeted by stuff like erythromycin. Oh, I'm doing this in blue when I was trying to do red. All the instructions in red. All right, so basically what these these antibiotics do, and then there's the 30S. I know these should both be red, but oh well. All right, and this is stuff like your tetracycline. So if you're on tetracyclines, pretty serious antibiotic, the tetracycline. You can't use this on pregnant moms, and you can't use it on children under 10 because it actually causes teeth staining. So you can only get this when you're an older person. Your teeth have basically stopped developing. So tetracycline is the other one, and this is going to target the smaller of the two ribosomes. And basically what it does is it prevents ribosomes from putting the amino acids together in the protein chains. I'm just going to put amino acids as AA, putting amino acids into protein chains. All right, so it prevents those protein chains from forming. Okay, and again, this is another static because it's not outright killing them like these two things are outrightly killed. It's just preventing them from being able to multiply properly or making the proteins for it to stay living or whatever it is. Okay, so basically that is it for your how antibiotics work. And again, if you want more information, you can just look stuff up anyway, but that's basically what I want you to know. All right, and again, you don't need to know the name of the drugs. I just think it's kind of interesting to learn about them. Alrighty, so that's it today. I'm going to save this as really low quality, as you probably are watching this, because that way I can load it up onto YouTube, and people who can't see videos because of you have crappy internet, or when the school website is overloaded, you can always see it. Alrighty? So make sure if you have any questions that you get a hold of me. Okay, thanks.